something that's really important in regulating the fluidity of our cell membranes is cholesterol. Remember that cholesterol is a steroid, so it's a lipid that is embedded into the cell membrane along with all of the proteins. So let's look at the picture. So here we have our steroid molecules, our cholesterol. Remember that steroids just kind of look like big loops? So that's our cholesterol right there. And cholesterol's job is to regulate membrane fluidity. So as temperatures rise inside of the body, cholesterol kind of sticks in between the phospholipids and prevents them from moving too quickly. Because remember how quickly phospholipids can move, 10 to the 7th times a second. We want to make sure that our cell membranes aren't turning into liquid and aren't like going crazy. So cholesterol helps keep them in their place. And then if it starts to get too cold, cholesterol molecules prevent them from freezing solid. So cholesterol is kind of like the temperature buffer for a cell membrane. So it's really important in regulating the fluidity of our cell membranes, whether it be too hot or too cold. It can work both ways. Another great example of structure-fitting function. So so there's the cholesterol, and here are the fatty acids versus, or the saturated fatty acids versus the unsaturated fatty acids. So here we have unsaturated fatty acids. They have double bonds in the hydrocarbon chains in the fatty acids, so you can see that they're bent. Because when you add the double bond, you bend the fatty acid. And so they can't get very close together because they're not straight. But once we saturate the fatty acids, we have all single bonds. They're in a perfect straight line. We can pack them really, really close together. So this one is more fluid. This one is more viscous. Viscous means thicker and doesn't run as easily. Like a pretty viscous liquid is peanut butter. Twice now it's come up in this video. So fluid would be like water. Viscous would be like a thick oil or like peanut butter or something. So saturated fatty acids are straight, and we get a very viscous cell membrane, whereas over here the fatty acids are unsaturated, they're bent, they can't pack together as tightly, they are more fluid. So, variations in lipid composition of cell membranes of many species appear to be adaptations to specific environmental conditions. So remember that we said that cell membranes are different between all different types of living things. And so since these cell membranes are all different, it's probably due to evolution and these living things evolving to adapt best to their environment. So a lot of plants change their membrane structure as temperature changes. So in the winter, when they don't want their cell membranes to freeze, they'll add more unsaturated fatty acids. And then in the summer, they don't want their cell membranes to be totally liquid, so they replace those fatty acid chains with more saturated fatty acids. So that's an evolutionary adaptation, which shows that these cell membranes have changed as all species have changed over time. So, membrane proteins and their functions. We talked a lot about the phospholipids. Now let's talk about the actual proteins, the other part of the cell membrane. And so there's tons and tons and tons of different proteins inside cell membrane. For example, a red blood cell has been found to have 50 different kinds of proteins on it. So that's a lot for something so tiny. And they perform basically all the functions of the cell membrane. The phospholipid doesn't really do too much. The proteins do it all. So they determine most of the membrane-specific functions. So the membrane of the Golgi apparatus is going to do something very different than the membrane of the whole cell. And that's one cell is going to do something, cell membrane is going to do something different if it's in your eyeball or if it's in your toenail, right? So they're going to be different, and this is determined by the type of proteins that we find there. So there's two main kinds. There's peripheral and integral. So if you think of peripheral is your side view vision. So peripheral proteins are stuck on the outside of the cell membrane. They do not actually go inside the hydrophobic layer of the cell membrane. Then we have integral proteins, which are integrated all the way into the cell membrane, and they do cross into the hydrophobic region of the cell membrane. So and the way that happens is because remember that different parts of the protein can be hydrophobic while other parts can be hydrophilic. So right here, this protein would have the hydrophilic regions on the outside of the cell and the hydrophobic regions in the middle. And the way that they do this is usually they have their secondary structure into a lot of alpha helices in the hydrophobic region that penetrates through the cell membrane. They often usually have channels also in them, so like holes right through the middle, kind of like a straw. That way things can pass through from one side of the cell membrane to the other. So these proteins are held in place either by the cytoskeleton on the inside of the cell membrane or by something called the extracellular matrix on the other side. Remember that extracellular means outside of the cell? It's called the extracellular matrix because it's like this crazy composition of all these different elements. And so it's the extracellular matrix outside of the cell. So let's talk about the 6 main functions of proteins that your textbook lists. So the first one is transport. This is very hydrophobic, this region in the middle, and so if something's not entirely hydrophobic, it's very difficult to get across. And something can't be entirely hydrophobic because then it would not exist inside the water, inside and outside of the cell. So, like I said before, a lot of these proteins have channels that go through the middle to allow things to cross. They're usually very specific, so you can see that only blue is crossing here because they only allow the blue thing through. We're going to talk a lot more about that later in this chapter. Sometimes these transport proteins actually change shape as they go through. So you can see that this one is open on this side. Well, it's going to open the other way to let it out on the other side. And that requires ATP. So we have to put in energy for that to happen. So that's transport. Then a lot of proteins that are stuck in the cell membrane, a lot of the integral proteins are enzymes. So they take one substrate and they turn it into another. Now, a lot of times they're kind of all stacked in a row on the cell membrane. That way, it's a pathway. So this goes in as red, it changes to blue because of this enzyme. Then this guy takes that blue and changes it to green. So it makes sense that they would be stuck next to each other in the cell membrane. That way, the different steps of the pathway can happen really rapidly. Then this one is called signal transduction, which means sending a message. So what happens is there's these signaling molecules. Like say this was a hormone. So the hormone is a chemical messenger that's coming from somewhere else inside the body. It comes to the cell and it wants to send a message to that cell. Let's say that this is insulin, and it's telling your red blood cells to take up more sugar. So insulin is going to send a message to this receptor protein on the cell membrane, and then that receptor is going to make a series of reactions happen to have something happen inside the cell. So this signaling molecule sent a message to the cell. Then we also have things for cell-to-cell recognition. So if we want two of our cells to communicate with each other, then they're going to need to be able to somehow realize what kind of cell the other one is. So this right here is a glycoprotein. Remember, a glycoprotein is a protein that has a carbohydrate attached to it. So this glycoprotein is going to tell this other cell, this protein on this other cell, what kind of cell it is, and then they can communicate. Sometimes we have two cells that are stuck together for a very particular reason. For example, in your stomach, your cells are stuck together very, very, very tightly because you don't want your stomach acid to leak out and melt your body. So it's important that those proteins can hold together your two cells. So intercellular joining is another reason. And that one says attachment to the cytoskeleton and extracellular matrix. So that's F on the figure in your textbook. And basically, remember, these proteins aren't moving around very well, and if two of them are stuck to each other, that way a pathway can happen. They are going to want to be attached. And let's say our cell wants to stay in one spot and not travel all over your whole entire body. Some cells want to do that, but some want to stay in one spot. These proteins can attach it to everything happening outside of the cell, right here, the extracellular matrix, and to the cytoskeleton on the inside of the cell. That way, the cell's not going anywhere. It's going to stay in one spot. And this is what I was just talking about, cell-to-cell recognition. The carbohydrates that are part of the membrane, usually attached as glycoproteins, sometimes as glycolipids, meaning a carbohydrate attached to a lipid. They allow the cells to communicate with each other. So they're on the extracellular side of the cell membrane, meaning outside of the cell, and the carbohydrates are covalently bonded to the lipids or the proteins, like I just said. So the kind of carbohydrates on the outside of your cell vary between species. So ours are going to be different than my dog's. They're also different between individuals. So mine are different than my sister's, are different than all of yours. And they're even different between cell types because my red blood cells are doing different jobs than my taste blood cells. So the carbohydrates attached on the outside are used to communicate with other cells, and they're different for all cells. We'll talk about that later. So the membranes have distinct inside and outside faces. So what's inside on the cellular side or the cytoplasmic side of the cell membrane is very different than what's on the extracellular side. The proteins are going to be different. The carbohydrates are going to be different. Everything is different. And this comes from backwards from the ER and the Golgi apparatus. So here we have the endomembrane system interacting entirely with the cell membrane. So we start off here. We have, this is the endoplasmic reticulum, and we see some secretory proteins. So these are proteins that are made by the cell that want to leave. So let's say this is a pancreatic cell making insulin. It needs to get the insulin out to the rest of the cell, or to the rest of the body. It's no good if it's stuck inside. So this is how it gets it out. So it starts off inside the ER. It leaves the ER in a transport vesicle, right? And so you can see that on this membrane right here, there are already transmembrane glycoproteins. So they're proteins with carbohydrates attached that are crossing the membrane. Transmembrane, across the membrane, glycoprotein. Protein with a carbohydrate attached. That's going to act as a signaling molecule. These little green guys are signaling or membrane glycolipids that want to be on the outside of the cell. So it's all inside here. This leaves the ER, comes to join with the Golgi apparatus. Golgi apparatus modifies a couple things, gets it ready to go. You can see it changed the shape of the membrane protein a little bit. And then this vesicle leaves the Golgi apparatus, and so the inside is this white layer. It's still on the inside. The red is still on the outside, inside the cell. But then once it fuses with the cell membrane, now the inside, the white layer, is the outside layer of the cell membrane, and the outside layer right here is the inside layer of the cell membrane. So it switches sides. So by implanting this glycoprotein on the inside of these vesicles, now they're on the outside, and they can communicate with the cells that this cell is going to be interacting with. So it's really important that you understand that the membranes have two sides. If you're on the inside of a vesicle while you're in the cytoplasm, once you fuse with the outside, you're going to be on the outside. So it changes as you join with the cell membrane, or if you're talking about just inside of the cell. And that is it for section 7.1.